Okay.
Yeah, can everybody now see my screen?
Yes.
Okay, then let's get started. First of all, I would like to thank the organizers for inviting me and giving me the opportunity to present some of our results today. Before we move into the control of biological systems with ultrasound, I would like to start with the field of optogenetics and photopharmacology. And as you can see on the left-hand side, optogenetics has had tremendous impact on the life sciences and medicine in recent years. Once you are able to control biological systems, for example, the control of protein function or the control of genetic circuits, then you are able to interrogate biological systems, selectively disturb them, and then you can learn about biological function and disease states. And in recent times, there were new therapies originating from these fields, and they were particularly exciting because they allowed to reestablish vision in blind patients. However, if you take a careful look at the animal here on the left-hand side undergoing an optogenetic experiment, you immediately recognize the big drawback of using light as a remote control stimulus. And the reason is associated to the very low penetration depths of photons into tissue. And that's why for many of these applications, as shown here, surgical interventions are needed. We offer an alternative, and the alternative trigger that we want to propose is ultrasound. Ultrasound has a very high penetration depth of more than 10 centimeters into tissue, and ultrasound is employed in the clinic for more than 5 decades, and this in a very safe way. That's also why ultrasound is already used in the field of drug delivery. And then with ultrasound, you can always exploit two effects. On the one hand, thermal effects and mechanical effects. And thermal effects in context of drug delivery have been exploited with, for example, thermoresponsive carrier systems, that once you induce a slight hyperthermia in the desired tissue part, the carrier releases the payload, and then you achieve the accumulation at the desired site. Similarly, you can use mechanical effects of ultrasound, and with this you are able to locally make the vasculature leaky. You can also open tight junctions, and you can even achieve the delivery over the blood-brain barrier. Similarly, thermal effects originating from ultrasound have been used for genetic control. In this case, scientists use thermosensitive transcription factors that allow the stimulation of or the expression of a certain gene in a target site. However, here, for example, in this paper, there were toxic effects due to the heating of the tissue observed. Another way, and again, now one exploits the mechanical properties of ultrasound. One can use mechanosensitive proteins. These are usually channels, and these channels, they can be opened by ultrasound. And then you have the influx of calcium. This can directly be used to induce nerve cell behavior, but it can also be combined with transcriptional regulators that are regulated by the concentration of calcium ions, and by this you can achieve the expression of a desired gene. However, if you look at the summary that I've given so far, there's no consensus strategy how you can, on a molecular level with ultrasound, selectively activate drugs, proteins, and genes. At the same time, there's a field in polymer chemistry that has developed over the past decade. It's called polymermechanochemistry. And in this research field, scientists, they induce also ultrasound as a tool to selectively break polymeric chains. The mechanism how this works is shown here. Once you apply ultrasound to a solution, you generate this cavitation bubbles. These cavitation bubbles grow, and once they reach a certain size, then they tend to collapse. And once they collapse, then there is a fluid flow towards the center of this collapsing cavitation bubble. And this flow can transfer shear forces on a polymer chain, stretch the polymer chain. If you then incorporate a mechanochemically weak bond, then you can achieve very selective breakage at the desired point within your polymeric chain. Here are some examples where this has been successfully performed. The mechanophores there is usually placed in the middle of the polymer because there the force actuation is most. Once you apply force or ultrasound, and then you can achieve, for example, homolytic bond cleavages, but you can also induce cycloreversion reactions at the side of the mechanophore. Another possibility is to incorporate aromatic moieties in the center of the polymeric chain. This is particularly exciting because it offers the opportunity to change the absorption and emission properties of the pi systems, and then you can use this type of aromatic chromophores as optical force probes within materials, and you can, for example, study the failure of the materials, and with this knowledge you can prepare probably more robust or more materials that break at desired force actuation. And before we entered the field, there were very few examples where upon the stimulation with force upon a polymer, small molecules were released. Before I enter the topic further, I would briefly spend a few minutes on explaining the different equipment that is used on the one hand in polymer mechanochemistry and on the other hand in the field of biomedicine. On the left-hand side, here you see a typical sonicator that is used in polymer mechanochemistry. It's a horn-type sonicator device that usually operates at low frequencies, typically 20 kilohertz, and which has a quite high intensities of 1 to 60 watt per square centimeter. I have to add at this point that these sonicators that are used in the field of polymer mechanochemistry, they are usually not compatible with cells and tissue. Here the intensity is too high, there's too much cavitation, and then you immediately destroy cells and tissue with this type of instruments. On the other hand, if one looks at medical devices, there are usually devices that are used for imaging, and one handheld device for point-of-care diagnostic is shown here, which we also apply in our lab. It's called Lumify. It's produced from Philips. They are usually operated at higher frequencies, and the intensities, as you can see here, are significantly lower than the ones for the polymer mechanochemistry devices. Besides these imaging devices, there are focused ultrasound systems used in the clinic. And here you divide between low-intensity focused ultrasound that has usually a low intensity, which is able to locally elevate the temperature slightly to around 42 and 43 degrees C. And then you have so-called high-intensity focused ultrasound devices. They usually operate at high intensities, but in this case, the aim is to cook tissue, for example, in the case of tumor ablation, and these devices are used in the clinic, but I would not call them really biocompatible with cells and tissue. So let's now take the opportunity and ask the question whether we can take these principles that have been developed in the field of polymer mechanochemistry and transfer them to the field of biomedicine and the life sciences, and to ask whether we can activate drugs by ultrasound. Here's one of the first experiments that we have performed. We have synthesized a synthetic polymer that contains an internal disulfide bond, and in the solution we have added a prodrug that can then be activated once you cleave the disulfide into the corresponding thiols, and then the thiols, they react with the prodrug and activate the prodrug into the active pharmaceutical ingredient. Here's the polymer that we have produced. Here you see the mechanophore. Here you see the prodrug that is, so to say, locked in the form of a Diels-Alder product. Once you, with ultrasound, you generate the thiol, then there is a reaction with the Diels-Alder product, and by this reaction, you generate or you introduce the retro-Diels-Alder reaction, and your active pharmaceutical ingredient is released. Here, for example, furosemide, which is an API that contains furan as part of its pharmacophore, but you can also release any other drug, for example, like doxorubicin, that in this case has been functionalized with furan to enable the prodrug formation and the activation by the thiols. The next step to evolve our systems further is to not only have the mechanophore and the prodrug in separate systems, but we wanted to combine them, and that's why we synthesized this second generation polymer systems. In this case, again, you see here's the mechanophore, the disulfide bond, but at the same time here, close to the disulfide bond, we have attached a carbonate linker, and on this carbonate linker now we can attach the corresponding drug. Again, if you now exert force on the system or you insonate it, then you generate the thiol. The thiol undergoes an intramolecular reaction with a carbonate linker. You form a 5-membered ring, but at the same time, you expel the active pharmaceutical ingredient, which in this case is camtothecin, which is a very hydrophobic anti-cancer drug. Here is now again the sonication shown. I have to add here and look at the symbol here. This is all done with the polymermechanochemistry ultrasound, so it's not compatible with cells and tissue. That's why we do a separate insonation experiment, and then we take the solution, we incubate HeLa cells with it, and then you see with increasing sonication time, we kill more and more of the HeLa cells, and the reason is because we liberate this anti-cancer drug that is cytotoxic to the cells. Besides liberating only a drug or a fluorescent molecule, we have also made a combination of that. In this case, you have now a system where with fluorescence you can monitor the drug release at the same time after ultrasonication. This we have achieved by making an asymmetric disulfide bond where then different payloads have been attached to both sides to the disulfide. Here you now see again the system. Here we have umbelliferone that is liberated as the fluorescent dye that turns on fluorescence once it's cleaved from the polymer by the mechanical force. And here we have a nucleoside analog that is also used as an anti-cancer drug. Here you can see only in the case of the polymer, once we have the polymer and we ultrasonicate it, then the fluorescence turns on, and at the same time the drug is released. And to really confirm that the liberation of our active payloads is due to mechanical force, we usually always do a control experiment where we synthesize the same polymer. However, now we put the mechanophore at one chain end where you have insufficient force transduction, and in this case you don't see any activation of the fluorophore, really telling that our activation or the bond cleavage is induced by mechanochemical principles. Now you may say the activation of a single or two drugs from a polymeric chain of probably 80 and 90 kilodalton is quite an insufficient process. This is correct, and that's why we also synthesized microgels that contain a higher drug payload. In this case, we incorporate the drug payload, then we sonicate the system. We also have incorporated the disulfide bonds as a part of the cross-linking moieties, and then upon the same principles as shown before, we liberate the drug. The big difference now is that we have an API content of 3 to 5 mol%, which is similar to micelles that are used in the field of drug delivery. We can release up to around 70% of the active payload. And another big difference and a big disadvantage is going to a different or going to the polymer colloids is that you need much less sonication time compared to the linear polymers. Here on the right-hand side, you see the different structure. The liberation is similar as shown before by inducing a Retro-Diels-Alder reaction induced by the thiols. And in this case, we have not liberated anti-cancer drug, but we have also demonstrated here that it is possible to liberate antibiotics. Let's move now from the cleavage of covalent bonds upon ultrasonication to the cleavage of supramolecular bonds for drug activation. In this case, we synthesize two types of gold nanoparticles that have a particular functionality on the surface. On the one hand, we synthesize small gold nanoparticles with vancomycin on the surface, and on the large gold nanoparticles, we connect beta-alanine motif that is, so to say, the target of the vancomycin usually on the surface of bacteria, but here we just use the d-alanine motif to inactivate the drug once you put together both types of nanoparticles. The nice thing about nanoparticles is that you can follow all the events by transmission electron microscopy. So here you see the large gold nanoparticles. You can even recognize the small peptide that we attach to the surface. Here you see the small gold nanoparticles with the vancomycin. Once we combine both systems, they form aggregates as expected. If you wait a bit more, the aggregates get larger, and then the drug is in the inactive state, and now we add the ultrasound, and now the aggregates, they get destroyed. You liberate the small gold nanoparticles with vancomycin on the surface, and they can then bind to the surface of Staphylococcus aureus, and upon the binding, they kill the drug. In this case, I have to add the Staphylococcus aureus. They are gram-positive bacteria, and they are very stable. So here you can do an in situ experiment. That means here you can use the Polymermechanochemistry ultrasound and have your biological system present, but this doesn't hold for mammalian cells, as shown before. Now the next question is, can we activate the activity of proteins with ultrasound? And for that purpose, you now need to do a bit of genetic engineering. What we do is we usually introduce unfolded polypeptide chains that are charged into a protein system. And the reason why you need to introduce this unfolded polypeptide chains is because you need to somehow a biophysical transducer that is able to transfer the shear forces from the solution onto the polymer scaffold. In this case, we have chosen green fluorescent protein, and subjected it to ultrasonication, and what now happens is that the 11th beta sheet that is connected to the rest 10 beta sheets is removed from the scaffold, and with this you switch off the fluorescent properties of GFP. So you can easily follow this over time. Here is a control experiment where we have just introduced a small linker here. And here we see that the protein is stable upon ultrasonication. However, if we put here the unfolded polypeptide chain that contains 36 negative charges, then you see that upon 10 minutes ultrasonication, almost half of the fluorescent proteins are switched off. If you now increase the length of the polypeptide chain with the negative charges, then upon 4 minutes of ultrasonication, you have almost switched off all the GFP molecules. In this case, we successfully realize the turn off of the activity of the protein, but you also want to use ultrasound to switch on protein activity. In this case, we have chosen for a similar design. We have taken trypsin, which is a well-characterized protease. We take here the one inhibitor for trypsin, which is BPTA. Again, you need to introduce an unfolded polypeptide chain by genetic engineering. In this case, it's a positively charged one that we have used. And now, once you ultrasonicate the system, then you pull out the inhibitor from the active site, and with this you can switch on the proteolytic activity of trypsin. On the right-hand side, you see the kinetic plots. Here you see the inhibited enzyme, and once you sonicate for 30 or 60 seconds, then you regain significant catalytic activity of the enzyme. And here the black is the control that shows the uninhibited activity of trypsin. So all the examples that I've shown you so far, they have been, let's say, controlled with the polymermechanochemistry ultrasound, and that means now the molecular principles, they have been established. However, the ultrasound modality that we are using is not really compatible with our desired system. And that's why we then put a lot of efforts into developing new technologies that allow to reduce ultrasound intensity for the liberation of bioactive payloads. And one of our platforms that we are working on are microbubbles. And here the idea is, once you have a microbubble, you have a core that consists of a gas. And this means these are very compressible colloids. And our hypothesis is, once you have a compressible core, you can actuate the surrounding polymer shell much better than by using the cavitation in solution. And with this, we hope to reduce ultrasound intensities. For that purpose, we have then made these soft colloids. We use microfluidics for that. We make the corresponding materials. They consist of polypropylene oxide crosslinkers, and as additional crosslinkers, we put in again the disulfide crosslinker that you already know from the previous slides. Here you see the bubbles that we produce. They are usually in the range, in a size range of around 25 to 60 micrometer. Here you see the bubbles floating on top of the solution after production. And here you see a SEM picture of the very homogeneous microbubbles. In this case, they are collapsed, but just due to the vacuum condition of the SEM. Then we performed the corresponding ultrasound experiments. And in solution, we again add a probe that can detect the formation of thiols. Similarly, again, by inducing the Rayleigh-Diels-Alder reaction as before. And now, again, we did first the ultrasonication with 20 kilohertz, and then depending on how soft we make our shells, we can activate around 20% of our mechanophores that have been incorporated into the bubble shell. And now I need to still owe you the proof that the hypothesis is correct. And for that purposes, we just replace the gas core with a liquid core by making double emulsions. Here you can see we make the double emulsions with the same size. We use identical ultrasonication conditions. And here you see no release or no activation of the disulfide at all. That at least proves that with using microbubbles, you can reduce the ultrasound intensities and achieve activation of your mechanophores. Then for sure, we also want to do chemistry in this microbubble shell. For that purpose, we synthesize here we not only incorporate the disulfide bond into the bubble shell, but we also incorporate here the profluorescent molecule, again by the Diels-Alder product. And now once you generate the thiol in the bubble shell after sonication, then you again induce the retro-Diels-Alder reaction and you liberate the fluorophore that now turns on its emission. In this case, I have to add here, we were able to do this with high intensity focused ultrasound. We developed another chemistry that now relies on the exchange of disulfide. So first you activate your mechanophore in the bubble shell and then you get disulfide exchange. Again, you form a 5-membered ring, but at the same time, you can liberate any payload that contains a hydroxy group as shown here for the fluorophore and for the camptothecene. And then a quite exciting observation was when we incorporate here directly a Diels-Alder product as the prime mechanophore in the system. Here we have taken a system from Boyston that was used and the furan was liberated by compression of macroscopic gels. However, in this case, the yield of the furan formation under compression was very, very low with around 3%. In our case, we incorporated a similar mechanophore in our bubble shell, and then we performed the ultrasonication experiment. And then we were very surprised to see that the efficiency of the process using microbubbles was much higher compared to the compression of macroscopic gels. In our case, we had a liberation of the payload of around 15%, which was significantly higher than once you use a macroscopic gel. So let's move now to the second platform with which we then achieve the lower intensity ultrasound activation of very different bioactive payloads. Let's start with the system. The system consists of high molecular mass polynucleic acids. This high molar mass was our hypothesis should be responsible for very low ultrasound intensities needed for activation of our bioactive payloads, because it was established before in the field of polymer mechanochemistry that the higher your molecular weight, or more precisely, the larger your contour length, the more efficient the mechanoactivation was. And for that purposes, we use this fabrication of nanoflowers by using a process called rolling circle amplification to make this high molecular mass polynucleic acids. So we take a circular template, we take a primer, we take the 4-nucleoside triphosphate, and then we add a polymerase, and this polymerase then runs more or less infinitely around these circles and produces a very, very high molecular mass polynucleic acid polymer. The cool thing about polymeric systems consisting of nucleic acids is that you are able to incorporate in binding sites, for example, for oligonucleotides, but you can also incorporate aptamer sequences that are able to bind small molecules or proteins. Once the payload are loaded on the carrier, then they are inactive. And now once you start sonication, then you release them and you transfer them into the active state. Let's start now with the activation of therapeutic nucleic acids. In this case, we have chosen for so-called CpG ODNs. These are short nucleic acid sequences that bind to toll-like receptor 9, and with this they activate in immune cells an immune cascade, and they lead to immune activation that can be, for example, exploited for vaccination, but it can also be exploited, as shown here on the slide, for cancer vaccination. Here you now see the nanoflowers that we have produced that contain the oligonucleotide payload. Here you see the sonication experiment, and now what you realize is that in this case we use the diagnostic device. So this is really now biocompatible ultrasound with a device that is routinely used in patients. Once we do the sonication experiment, we see that the size of the nanoflower decreases from around 2 micrometer to around 500 nanometer. And something that was very surprising to us after doing the experiment is that we not only liberate payload, as you can see, here's the oligonucleotide that is released from the carrier, but we also observe covalent bond cleavage of our polynucleic acid carrier upon ultrasonication. So here you see the control, that's the non-sonicated sample. The molecular weight is so high that the material cannot even penetrate the gel. However, after sonication, we see a smearing here, which really indicates that we have induced covalent bond cleavage with this imaging ultrasound. Then we tested our systems in vitro. First, we used the engineered cell line that contains just the toll-like receptor and reports on toll-like receptor binding by expressing enzyme that induces a color change once you add a certain substrate. And here just focus on the two important experimental columns here. When we just use the drug delivery system alone with the CpG loaded, we don't see any receptor binding. However, once we sonicate the system, then we see an increased receptor binding in this engineered cell line. And then we take macrophages. This is now an actual immune cell that contains toll-like receptor 9. And again, just compare the two bars here. There's no immunoactivation once you have no ultrasound present, but upon ultrasonication, then there's a strong upregulation of the cytokine release induced by the liberated CpG payload. And then the next step from a successful in vitro experiment, you go in vivo. So what we do is we take the DNA nanoflowers loaded with CpG, we tail vein inject them into mice, then we let the carrier accumulate for 1 hour in the animal, and then we insonate the liver for 15 minutes, and then we let the system run for 23 more hours, and then do the biological activation. And what we observe after this time is that the inflammatory mediators expression significantly went up. Here, this is the drug delivery system alone. This is the one where the liver was insonated. And here we have very high upregulation of TNF-alpha, interleukin-10, and CCL22 while in the interleukin beta type, there were other stimulation taking place just with the carrier alone. However, this experiment really tells that the polymers of the principles of polymer mechanochemistry can be really taken up into the animal and can be even employed in vivo. Besides using the ultrasound activation for immunostimulation, we were also able in vitro at least to knock down protein expression by delivering siRNA induced by ultrasound stimulation. For that purpose, you need to make two types of nanoflowers that bind the sense and the antisense siRNA. Once they are sonicated, the two oligonucleotides, they are liberated, they form then the corresponding bioactive moiety, and then we incubated the system. This is also done with the imaging ultrasound. We incubated GFP expressing cells, and by the liberation of the siRNA, you reduce the GFP expression. Here are the corresponding data. Important to show here is the siRNA alone without a transfection reagent is not potent at all. It's degraded and it's not taken up by the cells. Once you use lipoplexes, in this case, we have used a commercial system, you get a significant downregulation. Once we use the drug delivery system, we probably have a very slight downregulation, but upon ultrasonication, we really have a strong downregulation of GFP, showing that the system works very well. And now the striking advantage of using ultrasound compared to this highly cationic polymers is that the toxicity of our procedure employing nanoflowers is much less cell toxic compared to these polycations that are used in these polyplexes. So let's move on to the activation of proteins. In this case, we bind thrombin, or we load thrombin during the RCA process into our carrier, and then we liberate it. I don't show now all the experiments that we have done to characterizing the system with the 20 kilohertz ultrasound. I will immediately step forward to the experiments where we have used low intensity focused ultrasound. Here you see the experimental setup. This is our LIFU transducer. Here you have a multi-well plate, and with the focus we can directly address individual wells in this multi-well plate. And here you can see that we can that we have spatiotemporal control of switching on the protease activity of thrombin, as can be clearly seen here by the formation of the yellow color. And then we wanted to extend the system further. We don't only want to activate proteases, but with ultrasound, we want to be able to activate any type of protein. And that's why we use the thrombin released by ultrasound that I've shown you on the slide before. We combine this with two split intein units that you see here. Here you have the two units. They are connected with this thrombin cleavage site that you can see here. And once you liberate your thrombin, it will cut here and here. That means then you install the functional intein, and now there's an autocatalytic process taking place that cuts out the dark blue, the light blue, and the red blue part from the two extein units. And once this process happens, you connect these two units and you generate the functional protein. In this case, we have used this for the synthesis induced by ultrasound for fabricating GFP. But we also have realized this for the activation of calcitonin. In this case, our purpose was to use the ultrasonication to fabricate a bioactive polypeptide that is then able to downstream manipulate cell behavior. In this case, we have now taken the thrombin system, we have taken the split calcitonin intein system, and when we use the 8 megapascal focused ultrasound and the 17 megapascal focused ultrasound, we induce the formation of calcitonin. We all do this in situ in the presence of cells. And what happens then? Then you induce the influx of calcium in the cytosol, and you also induce the formation of cyclic AMP, which we can measure, and with this then control respectively cell behavior. Let's move on now from oligonucleotide activation, protein activation, to small molecule activation. In this case, the principle again is the same. We use an aptamer, load the nanoflowers with the respective small molecule. In this case, this is neomycin B. It's a very well-known antibiotic. Once we now incubate GFP producing cells with the nanoflowers that contain the neomycin B, and we do ultrasonication, then the small payload is released. It diffuses into the cells, and then it binds to a aptasine structure that is encoded in the mRNA. And once the small molecule binds here, you switch on the catalytic activity of this aptasine, and that means then your poly-A tail of the mRNA is cleaved off. That leads to instability of the mRNA, and by this, the GFP levels are downregulated. Here again, we have used focused ultrasound, the low-intensity focused ultrasound, and the imaging ultrasound, and for both of these, we see a very strong downregulation of gene expression, even higher here for the imaging ultrasound. The reason here is that once you have only a small focus in your multi-well plate, you have much worse sampling of your nanoflowers compared with this broadband transducer of this imaging device, and that's why we believe here we have even stronger downregulation. Yeah, with this, I'm at the end of my presentation, and I would conclude with a small summary. I've shown you that the principles of polymer mechanochemistry can be exploited to activate bioactives. The bioactives consist of small molecules, proteins, and nucleic acids. I've shown you that the microbubbles allow you to liberate small molecule APIs. With the polynucleic acids, we have a much broader range because we can liberate or activate all three classes of mediators. Important to mention, the ultrasound activation functions in vitro and in vivo, and evenly important for further applications is that the activation can be achieved by very low intensity ultrasound. And for that reason, I'm strongly convinced that sonopharmacology and sonogenetics enable new molecular tools to, on the one hand, treat diseases more efficiently and remote control biomolecular systems, overcoming some of the severe limitations of photopharmacology and optogenetics. I'm just a person presenting results. All the hard work is done by others, so I'm very, very grateful to the project leaders, to the postdocs, and the PhD students in my group. The ones that contributed to the work are shown here. I'm also thankful to the funding organizations that gave me the means to perform the research. And finally, I would like to thank you for your kind attention.